All right, good afternoon, everyone. It's a real treat to kick off this San Antonio Breast Cancer Symposium. Welcome all of you to San Antonio. If you're just traveling here for the very first time, it's an awesome place. My name's Reuben Harris. I moved here a couple years ago. I'm a professor at UT Health San Antonio, which is literally 12 miles northwest of here. So this session is on tumor heterogeneity and evolution. We have three awesome speakers. I'm just the warm-up act, so I'm going to try to keep it light. The rules are that we will ask questions at the end of them all. I think through your app you'll be able to vote, you'll be able to pose questions. We'll put them out there and we'll answer them in a ranked order, but the surefire way to have your question answered is to come to the microphone. Tell everybody who you are and share your question, and one at a time so that we can spread it out. Okay, so maybe a few of you know me, but I'm synonymous with Apobec. So years ago, we found these enzymes that deaminate cytosine to uracil, and my lab still works on their amazing properties of protecting us from virus infection, but also mutating tumor genomes. This is what I just mentioned, and that is the UT Health campus, if you haven't been there. So one of the things that struck me, I am not a MD scientist, many of you are, and some of you may even be pathologists. But when George Rees-Philo shared some pathology images with me of breast cancer years ago, I was struck by the fact that, number one, they're very pink and purple. But when you start looking at the details and you start looking for common features, it's actually hard to find common features. There's massive heterogeneity, even within subtypes like ductal carcinomas. And here he's listed all sorts of other types. But you know that many of you also know that this is not a single disease. So there's massive phenotypic heterogeneity in breast cancer. And so, of course, where does that phenotypic heterogeneity come from? And that's the kind of thing that we like to think about in my laboratory. So one of the sources, of course, is developmental trajectory. So if a cell of origin comes from one type or another within the breast, that'll dictate ultimate fate. Epigenetics definitely play a role. There's ongoing silencing and de-repression happening, and microenvironment, and we'll hear probably about all three of those themes at this meeting. But in this session, very little of that heterogeneity would happen without genetics, without mutation. And mutations, even in somatic cells, are heritable changes in DNA. We think about not just the mutation or the amino acid change that's affected, but the source of the mutation. So for mutations to happen, there has to be DNA damage. That DNA damage has to go unrepaired. DNA damage can come from inside the cell sources or endogenous sources, but it can also come from exogenous sources, like chemicals in our environments. And if it's ongoing, that allows evolution of the tumor cells from the earliest stages we'll hear to the very latest stages. It's not a static process. So heterogeneity, I want to use this slide to illustrate it. There's actually a massive amount of data. This was shared by John Martins in Holland. There's a massive amount of essentially whole genome sequencing data summarized on this slide, and it's 560 primary tumors worth of data up top and nearly 500 metastases down below. And the major mutational processes as defined by single base substitution number are indicated here: aging, mismatch repair, BRCA deficiency or homologous recombination repair deficiency, and APOBEC. APOBEC is in 2 shades of pink. BRCA HRD is in 2 shades of blue, and then you see this skiff over here. So aging is hydrolytic deamination of cytosine to uracil, mostly in CpG motifs. BRCA HRD, this single base substitution signature, is a surrogate for a lot of other events that are happening, larger scale events that are happening through the genome, and we'll see some of that in this session. And then APOBEC is scored as single base substitution, C to T and C to G in specific trinucleotide motifs. But another feature of these, and these are unrelated patients, is the fact that this component gets a lot smaller in the metastatic setting, so a lot of these were treated successfully, and the BRCA component nearly doubles, and the APOBEC component more than doubles. And that's consistent with these processes playing a role in ongoing evolution of those tumors. Another key point, which I think will come up later in the series of presentations, is the fact that these are largely mutually exclusive. Not totally, but largely. And that's likely due to the fact that they go down different evolutionary trajectories, and tumors don't require more than one process to fuel their evolvability. So we made this pie chart recently, and we didn't put water on here as a spontaneous source of endogenous damage because it would take over the pie. But the next biggest source across cancer is actually apobec, not just breast cancer, but all cancer. The next is HRD, reactive oxygen, tobacco, carcinogens, and UV light. Everything else that you could imagine is much smaller in proportion depending on the cancer type. So if these processes are ongoing, and I'm going to show some data of my own, but also some data from others, then they should contribute to the evolution of the tumors, including therapy resistance and metastasis. So these are early studies that we did with the ER-positive breast cancer cell line MCF7L, where we knocked down endogenous apobec and we slowed the emergence of resistance to tamoxifen. Over here, this is actually a recurrent estrogen receptor positive cohort that when these particular tumors recurred, tamoxifen was available as a monotherapy, and the apobec levels in the original tumor actually correlate with the outcomes in the metastatic setting. Okay, so low levels, much better outcomes than high levels. Here's another data set that you'll hear. Actually, this is courtesy of Avantika Gupta and Sarat Chandropati, and you'll hear about this in the main session on Thursday morning. But this is a couple of examples using the cell line T47D, where adding a little bit more apobec to that cell line can essentially give it the evolutionary power to overcome fulvestrant on the left, and on the right, the CDK4/6 inhibitor abemaciclib. Similar data with palbociclib, and that's just a little bit of the data that Dr. Gupta will be presenting Thursday morning. So that'll be a really exciting talk later in the meeting. Here's another. So this is not breast cancer, but I think it's amazingly striking. So it's a non-small cell lung cancer, and this slide is courtesy of Aaron Hata at Harvard MGH. And folks, there's still some seats on the right side if you want to go down that way, but standing is totally fine too. So this slide is from Aaron Hata, and essentially they sequenced not only the primary, but they sequenced multiple metastases, and each time this patient failed the lung cancer therapy, in this case crizotinib, I can't quite see it, I need better glasses, one of the other essentially tyrosine kinase inhibitors, resistance happened. And when you look at the actual mutations that conferred resistance, in all instances it's apobec. And moreover, the primary tumor barely showed any evidence for apobec mutagenesis. Most of the single nucleotide variations were from other sources, from aging and smoking. But after that tumor recurred, by far the dominant mutational process was apobec, and it clearly propelled it through resistance to multiple therapies. So I think similar things are likely to be happening with many of the amazing therapies that are available for breast cancer, but maybe not as striking with the numbers of metastases that were sequenced here. And so this apobec actually debuted at this meeting nearly 10 years ago, okay? It's hard to believe 10 years have passed already. And for those of you who haven't seen apobec in your practice or in your laboratory and are still wondering if this is a major mutational process or not, just by adding the two causative apobec enzymes to a murine system can cause cancer. So this is adding apobec3A or B by hydrodynamic injection through the tail vein into the liver. Large tumors, very large increases in tumor numbers in only a few months. If you make a transgenic that can be induced to just what we call the Goldilocks level of apobec, which actually recapitulates levels that we see in many different human tumors, we can accelerate tumorigenesis, and in this particular case, it's higher levels of apobec3B and liver and blood cancers. And we're working now to try to develop a mammary model of this particular apobec family member. So today, so this is just a little super brief introduction. We're going to have 3 awesome talks. The first is by Nick Navin, where he will tell us about actually a lot of these mutational processes, including, as you can see over here, changes in ploidy level happening even in normal cells before they become transformed into tumor tissue. The second talk will be by Dr. Christina Curtis. She'll talk to us about the genomics and the microenvironment and determinants in breast cancer progression. This beautiful figure may be shown again, where there's essentially from, if I'm understanding it correctly, from a diploid normalish cell, there's different evolutionary trajectories down here to this corner to triple-negative breast cancer and over here to ER-high, HER2-positive, with all sorts of associated phenotypes. And finally, Dr. Ashley Laughney, who will talk to us about single-cell genomics of triple-negative breast cancer. And the theme of this is how one particular protein is fueling many different phenotypes. So one protein, many functions in tumor development and progression. And last but not least, I still can't search the posters on my app. Maybe I'm doing something wrong, so I'm listing just a few. But if you search key phrases from this session, you will find many super cool posters. And I'm happy to say in almost all of the poster sessions, you'll find something that has to do with mutagenesis, chromosome variation, instability, inflammation, and ultimately heterogeneity. So it should be a super exciting meeting, and I'm really happy that this is a packed room right now. And there's still lots of seats, and I don't know why it says ADA seating, but people are welcome to use the front also if it's not taken, please. All right. Thank you. And just to remind everyone who's come in, we'll take questions at the end. We'll have a nice discussion. And the best way to get a question at the end of the 3 talks is come up to those microphones. All right. The first speaker of the session is Professor Navin.
All right, well, thank you so much and good afternoon. I'm excited to share some new data from our lab on the discovery of aneuploid cells in normal breast tissues and the implications for cancer progression. I have no disclosures to disclose. First, I just wanted to acknowledge some of the people that really contributed to this work. 2 really great graduate students, Ian Lin and Yunki Wang, who really led the study, both the experiments and the data science side. Kyler Wang, who was a postdoc, now has his own lab in Shanghai, who developed a lot of technologies. And the clinical side, we're very fortunate to have 2 wonderful collaborators, Bora Lim at MD Anderson and Alastair Thompson at Baylor, along with his team, which helped us obtain all these tissues that we used in this research. So I'd just like to start with our knowledge about karyotypes in normal tissues. The traditional view is really that most tissues have 22 balanced pairs of chromosomes or autosomes, along with your sex chromosomes. And we know this is to be true for most cell types, with a few exceptions, like things like megakaryocytes and some muscle cells. A long time ago, when we developed some of the first single-cell DNA sequencing methods, we had a few interesting observations. In one of the early studies we published looking at matched normal breast tissues, we found that there were a few cells in the matched normal area that had small deletions or gains of chromosomes. But these were usually kind of one-offs, and really this was more in the supplements of this paper. In a follow-up study in 2016, we looked more at matched normal tissues, and we estimated about 5% of the cells had these chromosome losses and gains, which we called pseudodiploid cells, because they're a little bit different than regular diploid cells. Now, there's been a lot of data coming out recently over the last few years, emerging data on mutations in normal tissues. This really started with Peter Campbell's work on the eyelids, showing that mutations can acquire over your lifetime due to UV exposure and can expand. And there's been a few other papers, including some really nice work from Roy and Lee, a new faculty that just joined our department, the body map papers looking at many organ systems and showing that mutations can expand, even driver mutations in normal tissues. So the questions we had for the study really were, do chromosome aberrations occur in epithelial cells of normal breast tissues, and do they expand, and do they have events that we associate with breast cancer progression? So I thought I'd start by quizzing the audience here. I'm going to show you the profiles of 2 cells here. The top cell has a gain of chromosome 1q, so that means it has 3 copies, and this includes the MDM4 gene, which suppresses p53, as well as loss of 16q. This is ECadherent or CDH1. And the other cell has that same 1q gain and also this loss of chromosome 10, which has the p10 tumor suppressor. So if I can get a vote, how many people think these are cancer cells? Few people? Okay, I gave it away with the title probably of my talk. But just keep these in mind as we go through the data that I'm going to present today. So we obtained 49 samples. These are reduction mammoplasties from women that are healthy with no known disease. A lot of these were collected as part of our Breast Atlas project, which took place over the last 7 years. We took the tissue, we dissociated it, and we made cellular suspensions, and we enriched them with EpCAM. And from that, we ran 2 different assays. One is high-throughput single-cell DNA sequencing. Another one is a new assay we developed in our lab that can do multimodal measurements, measuring both DNA and chromatin accessibility at the same time in those cells. We also took the tissue and embedded it and made blocks, both for H&E staining as well as for spatial transcriptomics, and using some more traditional methods like cytogenetics DNA fish. So this is the data from a single woman that I just want to walk you through very quickly. In this woman, you can see we sequenced about 1,649 cells. Most of them are diploid. They don't have copy number events. But if you look down here, there's about 93 cells that are aneuploid that have these chromosomal events. When we look at the tissue morphology, it looks normal by histopathology, the normal morphological and histological features in the lobules and in the ducts. And if you look at the cells, these 59 cells here, they had that gain on chromosome 1q and the loss over here in 16. And these other cells, there were fewer of these, but they had kind of chromosomal losses as well across the genome. And this woman was 44 years old. Here's another woman. She's 45 years old, and about 4.85% of her cells were aneuploid. You see the majority are diploid, as shown over here with all this white space, but about 105 down here were aneuploid. And they had things like chromosome 1q gain, loss of chromosome 10, which I told you is where P10 sits, and other cells that had kind of a few deletions as well. Again, the histology of the tissue was completely normal after being reviewed by several pathologists. So this is the data across all 49 women, and it's kind of sorted by the chromosomal events. And so if you look over here, you'll see that the gain of chromosome 1q is very common. In fact, almost most of the women have this event. We also see that chromosome 10q loss is very common, 16q loss. And there's a whole bunch of events over here that kind of occur at lower frequencies. We'll talk about those in just a minute. You can also see that chromosome X loss is very common, as well as chromosome 22q loss. Another way to look at this data here is by the frequency of those events. And now, in addition to showing you how common these are across the aneuploid cells, I'm also showing you whether cells with those events expanded clonally or not. And so you can see the events over here, 1q gain, X loss, 16q loss, 10q loss, 22q loss. Those tend to clonally expand within the tissues. Whereas you get down to this tail of other events, many of those are also kind of sporadic, shown here in blue. And so they represent kind of one-off events that happen. Now, we had a lot of clinical data we collected from these patients because they were part of our Breast Atlas project, and we did both multivariate and univariate analysis looking for associations. And the only thing we found, at least in the univariate analysis, that was statistically significant was correlation with age. We found with these women, both the number of CNA events as well as the proportion of aneuploid cells was positively correlated with increasing age. Now, when we look at the expanded events, we see that correlation is maintained, and it's true. But when we look at sporadic events, we see that correlation is not maintained. So in other words, those sporadic events don't seem to correlate with age. So now I'm showing you a frequency plot, and these are the normal aneuploid cells up here from normal breast tissues, and you can see those events that we just talked about, 1q gain, chromosome 10 loss, 16q loss, 22 loss, and X loss. And now I'm showing you the TCGA data from breast cancers. These are ER-positive breast cancers, and these are ER-negative. And you can see, of course, many of these events are shared with these invasive breast cancers, especially things like chromosome 10 loss. That 16q loss is very common in ER-positive disease, NF2 loss, and 1q is both an ER-positive and an ER-negative disease. Now, we looked at the co-correlation of these events, so we found that oftentimes when you have that 1q gain, it's accompanied by things like 10q loss or by 16q loss, and several of the other events were correlated together, as you can see here. We also did some deep sequencing of a few samples to look at allele-specific information. And here we were interested in whether the cells that have these events, did they really arise from just one cell in the normal breast tissue, or did they arise from different cells? And we found that in some cases that the 1q gains came from different alleles, the paternal or the maternal allele, suggesting that they arose independently and then kind of converged on expanding with these events. Now, we use another technology called Weill-DA. It's a nanowell DNA and attack profiling method to look both at DNA alterations and chromatin accessibility. And this is a new method from our lab that can profile thousands of cells. We have a patent that was filed on this, and we applied this to about 19 samples from this project. So with WellDA, we could then look at the lineages of the cells in the breast. Now, in the breast, we have these three main lineages: luminal hormone responsive, luminal secretory, so they form the lumen inside the ducts and the lobules. On the outside, we have the basal or the myoepithelial cells that are involved in contraction and producing that basement layer. When we look at the attack data, we can get these three clusters: myoepithelial cells, LumHR, and lum secretory, or luminal progenitors, they're called sometimes. And if we look at the copy number events in the aneuploid cells, they really occur in the two luminal populations where they're very abundant, and you rarely see them in that myoepithelial population, although there are a few. When we look at other events like X chromosome loss, we see that's very common in all the lineages. There isn't really kind of a bias towards one lineage. So now that we have this kind of dual data, co-assay data, we can look both at where the lineage of these cells are coming from along with their genomic profiles. And in some patients, we see that all those copy number events are really restricted to just one lineage, like luminal secretory. In other patients, we see that they're restricted just to luminal HR. But if we look at the majority of patients, we actually see that it's usually a mixture of two different things. We have a copy number profile, such as this one with loss of chromosome 22 here, that's associated with the LumHR lineage. And we also have luminal secretory cells that have a very different copy number profile. And these are intermixed in the same breast tissues. And this is another patient here where you see, again, the two lineages are represented, and they have very different copy number profiles. So the majority of patients, we actually see that it's a mixture of those two lineages, having different sets of copy number events that expanded. And in a few patients, we saw events in the basal myoepithelial cells, but there were very few copy number events in those lineages. So now we could take this data and look only at the normal aneuploid cells that were from the LumHR lineage and only the normal cells from the luminal secretory lineage. And we found that the ones from the luminal HR lineage matched much better to the ER-positive breast cancer data from TCGA, while the copy number profiles from the luminal secretory or progenitor lineage match much better to the ER-negative breast cancers. So this might suggest a cell of origin for these cancers. In the basal or myoepithelial cells, we saw very few aneuploid events, except for this loss on chromosome 3, which included the F-hit tumor suppressor gene, which we see more commonly in the ER-negative breast cancers. Now, because we have this attack data, we can also look at gene-specific events. And so we're very interested in genes on chromosome 1q in the LumHR cells. And we saw that chromatin accessibility changes occurred in things like MDM4, also on chromosome 16 for CDH1, and on 22 for NF2 tumor suppressor. In the luminal secretory cells, we saw MCL1 was significant, as well as MDM4. And then, of course, on chromosome 10, we saw chromatin changes in the P10 tumor suppressor gene. Now, we did spatial mapping as well, asking the question of where do these cells really exist if you look across all the normal breast tissue. And we can infer a copy number from the RNA recounts over here using spatial transcriptomics. And in this patient, we saw they localized this one lobular area over here. And when we looked at those cells, interestingly, we found that their morphologies, looking at the nuclear features and cellular features, they look completely normal. They didn't look like metaplasias, hyperplasias, DCIS, or anything like that. There was really nothing notable that we could see that was different about these cells. And we also confirmed these results using DNA FISH on the tissues. Here's another patient. This has just a 1q gain. We spatially mapped it to these little ducts over here in the tissue. And again, we see very normal morphology for the ducts, as shown over here, and the lobules over here in the tissue. So summarizing these results, we see that 40, oops, go back one. Thank you. There we go. Okay, so we saw in our results that 49 out of 49 of the healthy women in the study had aneuploid epithelial cells, and that the total number was about 3% of those cells that had aneuploid events. So judging from this data, we would expect that perhaps the majority of women in this audience here are likely to have aneuploid epithelial cells like this, and they increase with your age. We found that many of those aneuploid cells clonally expanded, about 88%, and that they really harbored events that we commonly associate with invasive breast cancers: 1q gain, 10q loss, 16q loss, 22q loss. They were mainly associated with the two luminal lineages, luminal secretory and luminal HR. And by the spatial data and histopathology, we see that they have normal cellular morphology and histology. Now, I wanted to mention another study that came out at the exact same date. We actually timed our studies to be published together. This is from Saurabh Shah, Sam Aparicio, and Joan Brugge. And they also looked at normal breast tissues as well as BRCA1, BRCA2 mutation carriers. And they came to a similar conclusion that about 3% of the cells were aneuploid in their tissues. And they also found that in the BRCA carriers that they had a few cells with lots and lots of aneuploidy, which our study did not look at. So we think this leads to a very interesting and provocative question of when is cancer really a cancer, especially for breast cancer. If we find these copy number events occurring in normal breast tissues and they expand with age, then what is the transition that really occurs to form those DCISs or premalignant lesions or invasive lesions? And so we think this is a really important question, and our data suggests that some of these events are occurring very, very early on in the normal tissue. And so I just want to thank all the incredible people from my lab that contributed to this work, our wonderful collaborators at Baylor, Alistair Thompson and his whole team, our cytogenetics core, Bora Lim as well, and our funding support. So I'll stop there. Thank you very much.
All right, excellent. Thank you, Dr. Navin. Our next speaker is Professor Christina Curtis from Stanford University.
All right, and yes, while I have a moment, I'll say there still are a few seats in the front if those of you standing feel like sitting. So thanks to Reuben and to Nick for, I think, setting the stage for some topics that I'll follow on from. These are my disclosures, none relevant to the talk. So it really goes without saying that tumors are dynamic ecosystems. They're fueled by the kinds of somatic alterations that we just heard about, but they're also sculpted by the microenvironment and certainly also influenced by our germline genomes. So we have a lot to work with here, but evolution will take place invariably. And of course, it's of interest to understand the rates at which that occurs, as well as the consequences. Now, we know as well that these somatic alterations can tell us about the history of a tumor and the relationship between cells, but it's also the case that I would put forth that they can provide future insights about what the tumor might do next and allow us to potentially predict disease progression. So today I'm going to walk through sort of three themes. First, I'll talk about modeling metastatic progression and the role that therapy has, because I think it's really important that we keep this in mind as we study breast tumorigenesis and progression. It's hard to decouple these two processes. I'll go on to talk about how subtype-specific mutational processes occur, akin to what Reuben alluded to in the beginning, and specifically to describe some new work that helps delineate the mechanisms by which these focal copy number aberrations arise and are propagated. And I'll go on to talk very briefly about how studying tumors, both their microenvironment as well as their genomes through neoadjuvant therapy, can tell us about predictive biomarkers. So we'll start here with this nice review paper from Kent Hunter, which really highlighted a question that plagued the field for some years and that we still seek to resolve, which is, how and when does metastasis arise? How is it specified? Is this a linear progression model? Do we have early dissemination with parallel evolution of subclones? Or do we have late dissemination of a single subclone? Are these all related? And finally, there's models where one would predict that there's just continuous but late dissemination from a variety of different clones. And of course, measuring this is challenging. We need both the resolution, but we also need the samples. And the way we've approached this is to leverage the fact that genomic sequencing data encodes the history of a tumor to trace back in time, at least to the most recent common ancestor, to the founding cell of a tumor. But we know that this information is really incomplete. And we use point mutations or somatic copy number alterations such as this to do this, but we also must simulate the process. And I would argue that simulating how a tumor grows is critical since we never have ground truth data on that process in the human system. And I won't get into the mathematics. I'll simply say that you can simulate three-dimensional tumors on the order of billions of cells. You can keep track of their spatial localization, sample from opposite sides of the tumor. And then we can use powerful techniques borrowed from the field of population genetics, which studies relationships between individuals applied to cells, and we can infer back parameters, such as the timing of metastatic seeding, the mutation rate, and so forth. And so in one example of this, we did this first in colorectal cancer, looking at paired primaries and metastases. And what we found using these kinds of computational modeling approaches that do account in the most naive sense for the microenvironment, we found that metastatic seeding can occur very early, long before we can potentially clinically detect the tumor. Now, this is obviously not in all cases. This is a biased analysis of those that metastasize, but this was provocative because it really suggests that some tumors are born to be bad, that their metastatic seeds are specified early. And we went on to then address whether this is true of other tumor types. So looking now at not only colorectal, but lung and breast, with a series of paired primaries and metastases, we asked this question. And of course, in breast cancer, the vast majority, some 83%, were treated. But using, again, mathematical modeling, we can come back, and what we find is that overall, the timing of metastasis is quite early, multiple years before we can potentially detect the primary tumor, and there's greater spread for breast, consistent with different rates of recurrence. Now, we can also ask questions about the mutations that drive this process. For example, we see that many shared events are present in the primary and the metastasis. These are known drivers, GATA3, TP53, PI3 kinase, but there's a subset that were unique to the metastasis. And in this case, what we found was that they are enriched for chromatin modifiers, and this was an analysis that really held true pan-cancer. So we're not seeing so many genomic alterations that are enriched, but mostly epigenetic changes. Now, you could go on and ask other questions, and in breast, we see that there's a larger number of metastatic-specific events or private events, again, very likely associated with treatment, and indeed, when we look across, you see that those cases that were treated have an enrichment relative to untreated paired metastases. So one way to think about this is back to the origins and the seeding of these clones, and you could predict either a monoclonal origin or a polyclonal origin where multiple cell clusters seed the metastasis. And what we see is that when we look at lymph node metastases that were untreated, collected at the time of initial diagnosis, they tend to show a higher degree of polyclonality. The distant untreated mets also show a higher degree relative to the treated ones. So this really suggests that we're selecting out for very specific events, and this is a lot of what we see when we look in the metastatic setting. So to summarize this part, really what we're finding is that the seeds of metastasis can be sown early, that many detectable private alterations are treatment associated, and that polyclonal seeding is probably more common than we can really pinpoint, and that is because treatment potently selects for the clone that wins, and it appears monoclonal. So this is really a backdrop for how we've now thought about addressing these questions in a more subtype-specific fashion. And some years ago, through the integration using machine learning on genome-wide transcriptional and copy number profiles, we described the integrative subtypes of breast cancer. You can see them here, 1 through 10. And what you can observe is that there are regions that are copy number amplified. For example, very much like HER2, which is amplified and overexpressed. We see other subgroups with these same characteristic patterns. You can see that the integrative subtypes help resolve some of the heterogeneity within the PAM50 or luminal, particularly amongst the luminal subgroups. And this gave us a platform for thinking about how we can further characterize breast cancer. So we see that there's 4 groups of ER positive, what we call high risk of relapse, 4 typical risk subgroups, 2 triple negative, and the HER2 positive group. Now, when we went on to look at the long-term follow-up of these cohorts, we're ordering them by their increasing risk of relapse. This is, of course, a cohort in which the HER2-positive patients did not yet receive trastuzumab. We have 20 years of follow-up. You can see they have the highest risk. But just adjacent to this are the ER-positive high-risk groups, with an excess risk of 45 to 55%. And you can see that this risk persists well beyond the time of surgery, 5 and 10 years later. Their risk is indeed higher than triple-negative breast cancer and the typical risk group. So we can summarize it this way, that there's amongst the 80% of ER-positive breast cancer, 25% have an exceedingly high risk. These account for the vast majority of recurrences, and you can see just the summary here, again, comparison with the HER2-positive group before the routine use of trastuzumab. These are the bad-acting tumors, and we need to better understand their origins and evolution. And this really leads me to where we've gone with this information, which is now, can we begin to parse what the underlying mutational processes are? Can we understand how these complex focal amplifications that look a lot like HER2 are generated? This required that we went back and assembled a cohort of whole genome sequence tumors. In this case, we've collected those that span pre-invasive ductal carcinoma in situ, invasive disease, as well as metastatic disease. And with this information, we can now go deep and start to parse not only the landscape of single nucleotide variants, but copy number alterations, structural variants, and complex rearrangements. So what do we see? Well, if we start looking just genome-wide, these are the copy number profiles, we can now use an unsupervised approach to parse what are those copy number signatures, what are the structural variant signatures, what are the mutational signatures. And if we simply project this in principal component space, you can see there's two main axes. We call this the architectural PCA, and each point represents a tumor profile. Now, if we color these, what you can begin to see is that there are extreme profiles, and they map in a way that allows us to see what is driving those subgroups of disease. So hopefully what you can appreciate from this is that clustered here on vertex 3 are the ER-positive high-risk tumors localized on top of the HER2-positive tumors. These focally amplified tumors have shared underlying mutational processes. We cannot discriminate between them when we look at them in this unbiased way. We can see the typical risk tumors up here and the triple negative enriched over here. So this allows us to take an unbiased view of what's happening. And if we map then the relapse potential of these tumors, looking at those that have a high risk of relapse beyond 5 years, you can see that they cluster down here in this group, particularly reflecting the ER-positive high-risk subgroups. We can also see that there are differences in their genomic features, and we'll drill into this. So I just want to come back to the fact that we've shown that these have specific copy number alterations, but until now we had not looked in depth at their structural variant landscape. What you can hopefully appreciate looking at chromosome 8, 11, 17 is that integrative cluster 6 harbors amplification and overexpression of FGFR1. This is a very focal event you see here. It looks quite similar to the amplification and overexpression we see for CCND1, and these are accompanied by other oncogenes that travel with it. And of course, down here we have the HER2-positive group. So while their oncogenes localize to different regions of the genome, their characteristics are remarkably similar. Now, it turns out that these ER-positive high-risk tumors, as well as the HER2-positive tumors, are enriched for cyclic or extra-chromosomal DNA elements. You can see that in the ER-positive HER2, the dark shading indicates that it's about 60% of these tumors. And in fact, the proportions are comparable in the primary versus metastatic setting, from blue to pink. So these two groups really dominate the landscape of where we see ecDNA, as we do see a slight increase, though, for the typical risk going to the metastatic setting. And importantly, these extra chromosomal DNAs encode many of those very specific oncogenes I just described to you that define integrative clusters 1, 2, 6, and 9. This is the darker shading, but they also co-occur with some other genes that travel along with them in those amplicons and on other chromosomes. So coming back to this, we've been intrigued, as have many others, as to how these events are generated. Now, there's been models proposed, one in which you can see that the estrogen receptor results in, we have induction of double-stranded breaks resulting in translocation events, and these dicentric chromosomes can form, resulting in chromosome bridges, where we get the separation and segregation of particular elements to different daughter cells. An alternate model, and many thanks to the work of Reuben and others, has shown that it's possible that estrogen receptor can in fact recruit ApoBec3b in a way that promotes double-stranded breaks. So we have the single-stranded DNA forming as basically a substrate for ApoBec. And this is occurring specifically at estrogen receptor binding sites where we have very high levels of transcription. Now, if we go back and look across primary metastatic samples, what we can see is that indeed there's an enrichment for these R loops that are specifically found at cyclic DNA regions above and beyond those in non-cyclic, and you'll see that the proportions are actually quite comparable between the primary and metastatic site. This would also give rise to a prediction that perhaps we would expect an enrichment of breakpoints that occur upon induction of estrogen signaling. So when we treat in a cell line, this is MCF7 treated with E2, you see that there's an enrichment of breakpoints that overlap specifically with the RAR alpha or SHANK2. So really what we're seeing is an enrichment for these. It is linked to estrogen receptor, and this perhaps provides a means for the genesis of these complex alterations. Now, we had the luxury of being able to look across disease stage. If we look in the ductal carcinoma in situ samples, we can see we can already detect these complex alterations. The high-risk ER-positive subgroups and their defining features are found. They're in fact present at roughly the same frequency as what we see in the invasive tumors. And I think this also harkens back to what we heard from Nick, that these alterations can occur early. In some cases, these tumors are addicted to those very oncogenes. And lo and behold, we see an enrichment of the ER-positive high risk of relapse subgroups in those patients that go on to ultimately metastasize. Now, these kinds of complex events are shown here. Here's a rearrangement that's specific. We see frequently in the integrative cluster 2 subgroup that fuses CCND1 and PAC1. These are two recurrently amplified regions on chromosome 11, Q13, to the FGFR1 locus on chromosome 8p11. So these occur, and they also occur in ductal carcinoma in situ. These very same events are found, and this is analogous to what we and others have reported in pre-malignant gastrointestinal lesions. So these alterations are there, they are defining features, and the tumors are addicted to them. Now, we can come back and parse the transcriptomic data. This is just bulk transcriptomics, and deconvolve them into the 4 TME subgroups that Bagayev et al. Defined. What you can see from this is that the ER-positive high risk and the HER2 show very concordant patterns. They're enriched for immune depletion. They do have a subset that show more immune-enriched fibrotic, shown in turquoise. And they are, really, as compared to the triple-negative subgroups, just immune devoid. Now, this also maps onto those 3 genomic archetypes that I already described. So we're really seeing that there's canalization of how the immune compartment interfaces with these mutational processes in a very stereotyped manner. And I think this simplifies some of the complexity despite these ongoing processes, and tells us that there might be distinct vulnerabilities in these tumors. Now, I'll come back to say that we also know that the microenvironments differ, and that intrinsic factors can influence immune evasion. So we went on to say, if we were to annotate all of the antigen presentation pathways, HLA class 1, HLA class 2, and looking at various epigenetic marks that influence immune evasion, where would these alterations lie? How would they distribute across subtype? What we can see from this is that structural variants in particular damage the interferon gamma and antigen presentation pathways. You can see that they're enriched here, as shown in purple. And in total, some 45% of cases show evidence for genetic immune escape mechanisms. These are hard-coded as a result of those alterations that I described. Some 9% are attributable to structural variants that we would miss without whole genome sequencing. We can also ask how subtype specific are these, so looking across the different integrative subtypes, what we can see is that certainly PDL1 is enriched, these alterations are enriched in the triple negative subgroups, but we also see examples where IDO1, which occurs on chromosome 8p11, is uniquely amplified in IC2 and IC6 tumors. So what we're finding is that the genomic archetypes can capture distinct mutational processes. We believe that looking at it in this manner allows us to better understand the underlying vulnerabilities. I haven't had a chance to drill into all of them, but these nominate new candidates that are particularly involved in replication stress, as well as homologous recombination deficiency. And so if we were to summarize, what we find is that the ER-positive typical risk tumors tend to have modest genome instability. They have higher levels of estrogen receptor signaling. They have modest immune enrichment. If we look at the ER-positive high risk and the HER2-positive, we see this early genesis of extrachromosomal DNA, focal amplifications that seem to drive disease progression. There's a reduction in estrogen receptor signaling, and immune enrichment is generally lower. And this contrasts with triple-negative disease, which has an overall higher level, but of non-specific genome instability. It is genome-wide, it is far less focal. And of course, these tumors are more immune-enriched, but that changes as they go on and progress to the metastatic stage. And so, of course, there's ongoing efforts to understand these new targets, particularly in the high risk of relapse populations. So to summarize this, really what we've shown is that the high-risk ER-positive and HER2-negative subgroups are driven by structural variants. These are coincident with those oncogenes we defined long ago using SNP-based array technology. They're frequently found on ecDNA, and this really suggests an evolutionary trade-off that I'll touch on just a bit more. And certainly now we're seeing evidence that estrogen receptor-induced R loops can fuel extrachromosomal DNA genesis, and that this is mediated by a process that once again implicates ApoBec. And finally, there are both genetic and non-genetic mechanisms of immune escape, and these differ between the subgroups of disease, and they differ in the timing in which they occur. So in just the last few minutes, I'll say that, of course, our broader goal is to understand tumor evolution in tissue context, to look beyond the genome and to understand the consequences on the microenvironment, to understand when immunoediting occurs and how this transpires, and really to have a holistic view that better models the complexity of the microenvironment. So we and others have wondered, we've spent a lot of time looking at the genome, but what happens to the TME as we look? And of course, the neoadjuvant setting provides an opportunity to do this in a very focused manner. And beyond that, we of course want to understand if there are molecular features that are in fact predictive of treatment benefit that might allow us to tailor therapy. So some years ago, I was fortunate to work with Sarah Hurwitz and Dennis Slayman. Sarah had led the TRIO USBO7 trial. This looked at trastuzumab or lapatinib or the combination in early stage HER2-positive breast cancer, and we know the drill. Trastuzumab was superior to lapatinib. The combination was not of great benefit. But what was hugely powerful about this cohort is that core biopsies were collected pre and on treatment before chemotherapy was administered, meaning that we can look after a single cycle of HER2-targeted therapy, what happened to these tumor cells. So together with Dr. Jennifer Caswell-Jin, we did a very deep dive into the molecular features based on bulk profiling, HER2-FISH, immunoscore, HER2-IHC, integrative subtype, intrinsic subtype. And what I can say from this is that very few were actually predictive of outcome. We went back and said, well, let's take another pass at this using newer technologies. And here we deployed a very early stage version of the NanoString digital spatial profiler, where we enriched for tumor regions using a pan-cytokeratin marker. We stained for 44 markers, protein markers, and we sampled a subset of these FFPE tissues through therapy. And really, this was a discovery effort, but what we learned was pretty striking. So if we just break this down by those patients who achieved a pathologic complete response, or PCR, versus those that didn't, you can see a dramatic reduction in HER2, phospho-S6, phospho-AKT, and many other markers. This is concomitant with an increase in a host of immune markers. We simply do not see this in the non-PCR cases. And we went back and looked in the bulk data. We still really couldn't recover this. So it could be protein, or it could be because we enriched for the tumor cells. But lo and behold, the result was compelling enough that we developed a digital pathology workflow to simply go in and enrich for these pan-CK positive regions and stain for one single marker, which was the most predictive in that model, and it was CD45, lymphocytes. You can see from the ROC curve that either the IHC-based approach for CD45 or the DSP gave us very high positive, gave us a very nice AUC of about 0.871, and in fact, the positive predictive value was 0.82. So this is quite good, and suggests that from this on-treatment biopsy, reading out lymphocyte infiltration might tell us which patients will achieve a PCR. And so we're in ongoing work validating this in a completed phase 3 trial, but the goal here would be then to say, could we de-escalate therapy in the subset of patients who we know will achieve a PCR? And of course, there's many efforts to do this. So I won't dwell on the other approaches, but to say, there's much more we can learn about the microenvironment in this context. We went back and looking at many other markers, what we can see is that there's an immune suppressive mark pre-treatment. We also see evidence for T cell exclusion. And if we look at the on-treatment time point, that hasn't changed a whole bunch. One single cycle of HER2-targeted therapy doesn't do it. But when we look by the time of post-treatment, we see that those signals are erased. And this really suggests there might be an opportune time to intervene with new therapies. It also emphasizes the role of trastuzumab as an immunotherapy. And of course, there's much more to be done about this. So we've gone deeper, and this trial has been the gift that keeps giving. We've now gone back and performed single-cell multiome, single-cell whole genome sequencing, and low-pass whole genome sequencing on subset of samples from this completed trial, looking longitudinally to say, can we better understand evolution in the context of a targeted therapy? And so I'll just focus very briefly on the bulk whole genome sequencing because I think it tells us something more about the mechanisms of evolution. So this is a landscape plot. These are individual patients over here, looking genome-wide at their chromosomes. You can see HER2. What I hope you can appreciate is there's actually a lot of variability in how the amplitude of the HER2 amplification event, as well as its breadth on chromosome 17. We can look at a single patient. This is trio 050. And what I hope you can see is there's a massive reduction by the time of baseline. So from baseline to run-in in the HER2 amplification. So single cycle of targeted therapy, we see those HER2 positive amplified cells go away, but we see its neighboring gene on chromosome 16 go up. And so the question is, over a single cycle, this is about 14 days of therapy, how can this happen? Well, one way would, again, be the genesis of these extra chromosomal events that are occurring in the context of potent treatment selective pressure. So we, in fact, can go back and reconstruct the kinds of alterations that we observe. This shows you the complexity of the HER2 locus. Now we're seeing other structural variants on CCND1. So again, cooperation is happening. There are many oncogenes involved that travel along with HER2. And we can look at this even deeper. So if we take primary tumors and just ask, which other amplifications are linked to structural variants in chromosome 17? You can see there's chromosome 8, 11, and so forth. This is what the chromosome 17 locus sort of looks like. And they're pretty well preserved in metastases. They're overall preserved. But we, in fact, can see that there's a whole new host of HER2 amplified focal events. This includes TERT, MDM4, et cetera, that only occur in the metastatic setting. So these may well be being selected for. And I would come back to the first point that I mentioned, that there is underlying evolution, and we have to look very deep, and we very likely have to look at the level of the whole genome. So just to say that HER2 might be a little bit different, I told you that it's similar, but HER2 has far more connected amplicons than any of these other co-amplified genes. And so, of course, it's of great interest to understand what that mechanism of cooperativity is. So in summary, we've shown that longitudinal profiling of on-treatment biopsies can reveal dynamic changes in the microenvironment, as well as the genome. This approach allows us to read out the potential benefits of treatment pretty early on, and certainly tells us something about how we might approach the next line of therapy. Clearly, tumors are dynamic and evolving systems, and they respond not only to treatment selective pressure, but to immune pressure. And so I think we have to think as a field about the opportunities to exploit this, as well as to anticipate resistance. So with that, I'll close and thank the many members of my team whose pictures you saw along the way, and our funding bodies, and thank you for your attention.
All right, excellent. Would anyone else like to come in and to come in and grab some seats? There's lots at the front. They're interspersed. Feel free, don't be shy. We're going to have an exciting Q&A after this. But our next speaker is Ashley Laughney, who's a professor, assistant professor at Weill Cornell Medicine, and she's got an exciting story to add to the bunch and the discussion. Thank you.
Okay, thank you for the opportunity to present. So today I'm going to actually talk about another source of heterogeneity that tumor cells can exploit. Is this? I'll use the mouse, I guess. Okay. And that is pleiotropy. So one gene can give rise to many functions, some that can help a tumor progress and some that can restrict it. And so if we want to effectively target these pathways in cancer, we need to understand their context-dependent functions. And so this is really a great area to explore with systems biology methods. So as a whole, my lab focuses on tackling this genotype to phenotype problem, predicting protein function from sequence and complex evolving systems that are multicellular. And so that system for us, of course, is the metastatic cascade. This is a very simplified version of this tumor progression and metastasis, but what I want to emphasize here is the biology in primary tumors is very different from the biology in metastasis. And even within the metastatic cascade, we know there's very different biologies in more dormant or latent metastases as compared to more aggressive models. And so this is very important for us to keep in mind. Over the years, we've learned that there are a lot of parallels between an injury response and tumor progression. We think a lot about metastasis specifically as a non-healing wound that triggers both chronic inflammatory signaling, which then leads to both immune remodeling and regenerative programs that ultimately trigger growth at a distant site. Well, this is an old concept. We think about it from a new perspective in that we don't think about the tumor physically disrupting its surrounding epithelium, but rather we think of instability, this ongoing process of chromosome missegregation events, as a driver of that chronic inflammatory signaling. And so we've already had a great preview to this, but I just want to highlight that most aggressive metastatic cancers exhibit profuse copy number alterations. So here you're looking at sky karyotyping. Each chromosome is painted a different color. These are individual cells all sampled from the same tumor, and what you can appreciate is no two karyotypes are the same. And so one source of these copy number alterations is an ongoing process called chromosomal instability. And it's important to recognize that this is a process, it's a rate, an ongoing process that is distinct from the state of abnormal copy numbers. And so beyond just thinking about changes in gene dosage, I want to encourage you to also think about the transcriptional response to this this ongoing process. And so it turns out these aggressive metastatic cells frequently, they have defects in the fidelity of their whole chromosome missegregation. They often generate these lagging chromosomes that erroneously end up in and I just want to emphasize, we typically say that there is no genomic driver of metastasis, but in fact, fraction genome altered is consistently enriched and matched metastases as compared to their primary tumors in more than 25,000 patients that were assayed at Sloan Kettering. And so there is a genomic driver of metastasis. It's just not one gene, it's a process. And so it turns out those lagging chromosomes end up frequently in these very fragile rupture-prone micronuclei. And when those micronuclei rupture, they expose DNA to the cytosol that actually triggers an innate immune response chronically, because these missegregation events happen every 2 to 5 cell divisions. And so this is really an area that our lab focuses on. What are the consequences of this chronic sensing of cytosolic DNA? Several years ago, we contributed to the discovery that when you we decoupled the state of aneuploidy from the rate of missegregation. So we took otherwise aneuploid, in this case triple-negative models of breast cancer, and we increased or decreased their rate of missegregation by modifying kinesin 13 protein family members, and we showed definitively that this ongoing process, by increasing the rate of instability, we potently drive metastasis. The unexpected finding, of course, was that this depended on STING, this innate immune sensor. Since then, we have shown in fully immune-competent models as well that indeed, when you knock out STING, you suppress metastasis in these chromosomally unstable cancers. And when you add back STING in a dose-dependent manner, you get a dose-dependent increase in metastasis. Moreover, not only did this hold true in the fully immune-competent setting, but it actually required interactions with the immune system. So if you modulate SYN or downstream STING activation, and the fully immune-competent setting, you see a STING-dependent metastatic effect, but that goes away in the NSG mouse that is severely immune-compromised. And so, of course, this begs the question, how is SYN and chronic activation of STING reshaping the tumor microenvironment to switch this otherwise very immune stimulatory pathway on its head? Why is it now promoting immune tolerance and progression? And so to tackle this, we developed a method called contact tracing, which I'll talk very briefly about. Melissa Hubitz led the development of that method, and this project was a joint collaboration with the Bakum Lab, Sam Bakum, who's now at Velastra, and June Lee, who's a very talented postdoc. And so how do we understand the effect of cell-cell interactions in the tumor microenvironment? Typically, when we infer interactions from single-cell data, we're looking for complementary expression of ligand receptor pairs. And there are many databases that are assembled in very smart ways to annotate these complementary ligand receptor pairs. If both are mutually expressed, bing, 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 putative interaction. The next wave of methods said, you know what, let's prioritize interactions that show evidence of downstream signaling in the target cell. So when that target cell type binds a ligand to its receptor, it should change its behavior. This was a great concept and a conceptual advance. The limitation was that this evidence of downstream signaling was based on very limited prior knowledge, where basically they took a panel of epithelial cell lines. This was done at the Broad, the CCLE database. They would treat them with various ligands, sequence before and after ligand addition, a targeted panel of, I think, 1,000 genes, and say this is what response to ligand A looks like. The two limitations of this approach are, of course, that these are responses measured in a dish, which often do not reflect the responses we see in complex in vivo systems. And two, they were cell type agnostic. So it assumes that the downstream response to binding to a given ligand is the same across T cells, B cells, tumor cells, NK cells. And these are different cell types for a reason. They respond to stimuli differently. They have different chromatin accessibility, et cetera. And so we had a very simple idea, which was, can we actually use concepts from variational inference to infer how a cell responds to a given ligand receptor mediated interaction from the data itself in its native in vivo context? So if there was, for example, an interaction predicted between a tumor cell secreting a ligand that binds to a receptor on a macrophage, we would say, okay, well, not all of our macrophages are expressing the receptor, and not every microenvironment has ligand available. So by exploiting these two key sources of variants in a biologically inspired way, we can actually tease apart genes that are putatively causally induced by the ligand from those that are just simply correlated with, I talk with my hands too much, with receptor expression. And so all of the source code is available online. This was published now over a year ago on Nature, where it was at length validated both the predicted response genes as well as co-localization of our predicted interaction, showing that we prioritize interactions in a way that best captures what's happening with the biology. And so this is a very simplified output. It's called a circus plot that basically highlights, we focus on any interaction in the microenvironment that measurably shapes the target cell in the TME. So if that target cell changes its behavior, which is encoded in the thickness of this ribbon, and we look at differential abundance of the ligand as well, and how that depends on both our perturbation of SYN or STING. And so if we zoom in on the ligands that are emanating from cancer cells, measurably shaping their targets in the TME, we really interestingly discovered that many of those ligands were associated with an ER stress response. Conversely, if we took those same tumors and we either suppressed rates of instability or knocked down STING signaling, now many of the ligands emanating from the cancer cells were associated with more of a type 1 interferon response. And so this was intriguing to us because while we typically think of STING and interferon as synonymous, ER stress is actually an evolutionary conserved function of STING. And so we asked, could there be a STING-dependent ER stress response in our tumor cells? And indeed there was. So here we've induced ER stress chemically with tunicamycin. TMEM173 is the old gene symbol for STING. I should update that. But you can see that indeed there is a STING-dependent ER stress response in these cancer cells with SYN. And if we knock out any one of the three arms that mediate an unfolded protein response to ER stress, we significantly suppress metastasis in these chromosomally unstable models, again, and as predicted, in a manner dependent on interactions with the immune system. So how does this evolve? We had a hunch that it had to do with chronicity of activation because SYN is such an ongoing process. But to understand dysregulation from an already dysregulated state is very challenging. So we actually took a reductionist approach. We took fibroblasts that are mostly diploid. They do not have chronic activation of STING. And we either treated them once with the STING agonist CGAMP or chronically. We would treat them daily, in this case for up to 5 days. And what you can see is after one treatment of CGAMP, you get a robust interferon response. This is 200-fold induction of interferon beta. But by the 5th daily treatment of CGAMP, these cells they've become desensitized and they no longer want to produce interferon. Conversely, you see that with chronic activation, you begin to see an upregulation in a lot of these chaperone proteins that mediate this unfolded protein response to ER stress. So by virtue of just the chronicity of activation, we can flip the functional state of STING from an anti-tumor interferon response to a pro-tumor ER stress response. And so, I think STING is a really beautiful example of pleiotropy and biological systems. And so it's really important that we understand these adaptive functions. When are they activated? Should we be activating STING, inhibiting STING? This is a really complex question. And I just want to point out, too, we observed when we chronically activated both the fibroblasts or in cancer cells when we inhibited chronic sensing of cytosolic DNA by knocking out CGAS, we saw rebounds in STING expression levels. So I just like to point out that there is when STING is activated, it actually triggers its own degradation via autophagy. We had to remind ourselves of this. This was actually known. And so if you inhibit autophagy with baflomycin, you can actually prevent that degradation. So in patients, how does this manifest? I think this is important. If cells are primed to mount that anti-tumor interferon response, they're actually going to have high levels of STING and low levels of CGAS. But if this pathway is chronically activated, typically you see a starry sky. These are all ruptured micronuclei that are recognized by CGAS. And in those regions of ruptured micronuclei, you're going to see lower levels of STING protein because it's actively being degraded. No cell wants to constitutively produce interferon. It's very stressful. And so this led us to ask, because obviously STING has complex effects in both the tumor and host, can we inhibit STING itself pharmacologically in tumors with chronic activation of this pathway, and will that extend survival? And so here the answer is yes, and we showed that the effect depends on tumor intrinsic levels of STING. And so how do proteins evolve multiple functions without messing up their old functions that they needed? One way is through the evolution of distinct modular domains, and STING is no exception. Actually, its ability to mount an interferon response is entirely encoded in its C-terminal tail that really only emerged in vertebrates, which is very interesting. And so, we know that one protein has many functions. This is a problem for many reasons. One, it, confuses us about whether we should be activating or inhibiting a protein when developing targeted therapies for cancer. Also, typically when we think about developing a drug, we're often thinking about all or none drug effects, where really we would love to be able to stratify patients in those that we should activate STING, for instance, or inhibit STING, or even better, could we just inhibit that pro-tumor ER stress function of STING while enabling its interferon response? Wouldn't that be amazing? And so the goal of this next project, which is unpublished, so please don't post this, but we wanted to ask, can we map which parts of the protein contribute to each of STING's functions? Are those functions independently regulated by different residues? And can we inhibit specific functions of a protein, these separation of function drugs? And so, this is a model of the STING homodimer, and the approach is very simple. We basically take every building block of this protein. So for human STING, there's 392 amino acids. For each position, we mutate that residue into every conceivable counterpart. So this is deep saturation mutagenesis, and we basically march along the protein and systematically substitute every residue with every other conceivable counterpart. And we do this in a way where we actually couple every mutation with 2 DNA barcodes, one that tells us where the mutation was introduced, and then with long-read sequencing, the second mutation tells us, the second barcode tells us what substitution was made at that site. We've designed these barcodes to be captured by your favorites, scalable, 10x, what have you, single-cell approaches. So we can readily, in a one-shot experiment, map the function of every single residue of a protein, which mostly exhibit loss of function, although you can find occasional gain of function mutations. And so then the workflow is to deconvolve and map those protein functions. So you compare the gene expression of every mutant to the full-length wild-type protein here of STING. We can assign those differentially expressed genes to functions, map them to the 3D structure, and then look at, well, when are those functions important at different stages of disease progression, and how do those mutations interact with drugs that exist to target this protein? And so this technology is pioneered by a very talented postdoc in the lab, Matt Yale, and is being applied to STING signaling in cancer by an equally talented graduate student, Amelia Owenstad. And so where do we fall in the functional genomics landscape? Obviously we could compare each variant to the full-length protein and do deep sequencing one by one. This is not very scalable. Saturation mutagenesis is at the other extreme, where you can look at every possible variation of a protein, even those that are not observed in patients. But your readouts are typically one-dimensional. You need to know what function you have to assay, and your measurements are all biased towards that measurement function. Whereas SABCS is positioned to assay genome-wide in an unbiased manner the function of every single perturbation of this protein, which requires no prior knowledge about this, although we do think a lot about STING, and can readily be scaled to other targets. It is significantly more efficient than base and prime editing, which I'm sure is making great strides and often is used to look at patient-observed mutations in cancer, but still efficiencies like, around 20, 30%. And so basically to start, we wanted to capture loss-of-function mutations of STING. We do this again in a cellular context. We've even measured these transcriptomes from cells isolated from growing tumors in vivo. And so we can look at the response of every variant, barcoded variant, to CGAMP stimulation, the STING agonist. We can classify actually just based on the full-length protein, and also we've spiked in stop variants that encode a stop codon that do not produce protein to precisely train a classifier to say this is what a response to CGAMP should look like. And then we see that many of the genes that define that response make a lot of sense. They're a lot of our favorite interferon-stimulated genes. But we can identify other loss-of-function variants that really don't behave how they should when stimulated with CGAMP. Some of these are known, we can look at the genes that are differentially respond to CGAMP in those loss-of-function, and some are novel. Just to kind of show you high level, like here in the stop mutation, we see loss of canonical functions of STING, including interferon, NF-kappa B, unfolded protein response. If you chop off the C-terminal tail, now you specifically see loss of the interferon response instead of NF-kappa B signaling pathway. Mutations, this RRDD mutation is known to be a loss of UPR function. What I've been surprised to learn is actually a lot of these loss of function mutations are not as specific as we think they are. There is a lot of promiscuity and crosstalk between the functions. And we're now in the process of actually functionally validating novel loss and gain of function pathways variants by subcloning these out and validating these in vitro. And we can map them to the 3D structure of the protein as well to try to understand. To map. And preliminarily, it really doesn't seem like there are major, large domains that determine key functions of STING. So we actually think a lot of it has to do with who they bind to at those sites. We can then take our complete barcoded mutagenesis library and we can assay it in vivo and say, when are these different functions of STING important? So we've done multiple in vivo assays, including mammary fat pad injection, and we look at both metastasis to the lung. This is in the 4T1 model. We've also done tail vein assays. This is an example in the EO771 model. These are both triple negative models of breast cancer. EO771 is interesting in that it can still mount an interferon response. 4T1 has totally lost this. And so what we see, this was one of our first experiments with the full library. We see that if you mutate residues responsible for the ER stress function of STING, the pro-metastatic effects go away. And the highest or the most pro-metastatic variant detected, the greatest change in gene expression was related to loss of interferon, which was really nice to see. This is an example of both primary tumor fitness and metastatic fitness in the 4T1 model. And what I want you to appreciate is that the fitness effects are much larger. So the axis is almost double here in scale compared to the primary tumor. It's much larger in the metastatic setting as compared to the primary tumor, at least in this chromosomally unstable model of cancer. And this is in line with what we've observed. If we knock out STING in primary tumors, it really doesn't impact tumor growth in these models, which again are very chromosomally unstable. But if we knock out STING, of course, that suppresses metastasis. We've assayed whether, the metastatic fitness is explained by changes in growth. It's not. We focus on variants that are conserved in humans, and then we've actually started looking at conservation of these residues across at least mammalian species and whether mutations have been observed to narrow in our scope. We are focusing on a particular lysine here at position 337 right now. That is the most metastatic variant in multiple experiments. And we see that this site is a known site for TRIM29-mediated polyubiquitination, and as well as sumoylation. And so this likely is responsible for stabilization of STING. And so we're following up on that now. And so context is key. I just want to highlight too here, we've clustered all of the fitness across positions of the protein across assays. This is just for the 4T1 data, but you can see that the metastasis and tail vein experiments more closely cluster together than primary tumors, and those cluster all distinctly from growth rates in vitro. So then we wanted to ask, can we actually predict now how different drugs interact with these functional elements? Because that would be pretty amazing. And we thought, and this was kind of an afterthought after we made this library, we know drugs can act through on and off target mechanisms. If there's an off-target effect, it should affect all of the variants uniformly. But if there's an on-target effect and you mutate the site at or near where the drug binds, that drug effect should go away. And so we wanted to see if this was possible. And so here I'm just showing distance in gene space, Euclidean distance across all highly variable genes for every variant in our STING library before and after treatment with a STING agonist called DMX-AA. This was the first agonist used in clinical trials, and I chose it for a specific reason, which I'll get to in a minute. But you can see that mutants that encode the stop protein, or that do not produce protein, they introduce a stop codon, show very little drug response. Our full-length wild-type protein exhibits a robust gene expression change in response to the drug. This drug, this agonist, actually failed in clinical trials because of a single amino acid substitution difference between mouse and human STING at the drug binding site. Where was that? Here at position 229. And you can see from our in vitro assay at a bench, we could readily predict that resistance mutation. Additionally, a year after that resistance mutation was described, another mutation was described, and they said, hey, actually, if patients carry a mutation of this serine at position 161, then they will respond to DMX-AA, which otherwise only works in mice. And you can see where that responds here. So now we have a quick tool, once you have your library, that we can now apply to all sorts of STING activators and inhibitors to say who's likely to be hyper-responsive, who's likely to be resistant to these drugs. And I want to point out, we've also validated some novel hyperactivating and inactivating sites. And I want to point out this is not a cytotoxic drug. If it was a cytotoxic drug, we could just select the variant library and drug and sequence what survives. This drug acts through interactions with the immune system. So it's not killing the cells, and this is a great way for us to identify those. And so in some I've told you about how STING can give rise to many functions. We talked about how chronicity of its activation can flip it from an anti-tumor interferon response to a pro-tumor ER stress response that leads to immune suppression. And we're very excited to keep moving forward with SABCS to basically deconvolve these different protein functions to different parts of the protein and hopefully target those specific protein domains. The technology is also very useful to identify both resistance and hypersensitizing mutations and better inform drug binding sites, especially for highly disordered regions of a protein. If you don't know where your drug binds, this might be a ready way to figure that out. And finally, of course, I'm super privileged to work with this amazing team of scientists who I would like to acknowledge here, our funding sources, and I'm all done. I guess we'll take questions as a group. Thank you.
All right, that was fantastic. I want to thank all three speakers again. That was really great. Please come if you'd like to sit down. We'll take the first few questions on the microphone. I was actually learning how to use the app in real time as we were going along, and you do not have to log into the session to have a question. You can, or to vote on a question that you want to have someone ask, okay? So you can just go in there and hit Q&A, and then you can give it a thumbs up or a thumbs down to elevate it on our list of electronic questions, okay? So if I can figure it out, you guys can definitely figure it out. All right, maybe question one, question two. Question one over here.
First question for Nick. Great talk on the Aneuploid. I just wonder, have you looked at other stromal cells in the breast in addition to epithelial cells? Are those specific to epithelial cells, or could it be commonly seen in other stromal cells like fibroblasts and immune cells?
Thank you, great question. Although in the paper we focus mostly on the epithelial cells because we enriched with EpCAM, we have done experiments just looking at all cell types, and the majority of these types of cells I'm showing you are epithelial specific. So.
Okay, thanks. Second question for both Nick and Christine. You have talked about the copy number change in both potentially tumor development and metastases. How do you think that may interplay with tumor plasticity, or whether those be more predominantly in stem cells? For example, they will actually be playing a role to both tumor development and metastases.
Yeah, I mean, it's a great question. I think there's multiple processes that are operative here. There's been really beautiful work, including from Ashley and many of her colleagues, showing the role of cellular plasticity in this. I think we didn't look at that explicitly in the study I described, but I think, it's operative. And the point that I made in the beginning about the role of these, certainly we do see some enrichment for epigenetic chromatin modifiers would be consistent with that. They don't have to be mutations, of course, and we're missing a lot potentially by not looking at epigenetic changes. We can read some of that out transcriptionally. So, unfortunately, I didn't have time to talk about some of what we see there when we do use ATAC-seq and other methods, but you can start to see some of those cell state shifts, for example. So, yeah, great question.
Super. This microphone. I'll call it 1, 2, 3, and 4.
Diako Ibrahimi from Texas Biomed. I have a question about the STING function. So you are seeing two different functions for STING depending on the context, metastatic tumor versus primary, and you look for sources of this functional difference inside, just I'm guessing, a reference STING structure. Is it possible that within primary and metastasis, STING produces different protein, perhaps different splice variants?
So STING is very rarely mutated in cancer. I don't think it's a necessarily different variants. I think it has to do with like chronicity of activation. And also just there's a lot of changes in calcium flux in the cells. STING is an ER resident protein. It has a lot of different binding partners. And where STING is, I think, is really a very important determinant of what STING does as well. So, yeah.
And I'm going to bounce into the E questions for just a minute for Dr. Laughney. So many are curious, how do you assay 7,500 protein variants simultaneously? So
Great
it must,
question.
I'm going to guess it's barcoded and into a null cell, but I don't know.
Yeah, so we have two barcodes, and one that says where the mutation was introduced, and a second clonal barcode that then we can use to map what substitution was made with the long-read sequencing. So we are doing, some pretty big single-cell experiments to cover them. We do often, a lot of the assays I do often collapse per position to increase cell power. So that is, we are, the single-cell experiments can be very expensive to scale up for the full library, although it's feasible. So we collapse per cell often, and we do look at whether or not multiple clones can contribute to a given phenotype, or if it's clonal. And if it's clonal, then we'll say, okay, well, why is it clonal? Is it a specific substitution only at that site that gives rise to?
And I assume also that the library is transduced in and selected so that every cell has at least one of those variants.
Yes, so we transduce with a very low MOI. We're very cognizant of recombination effects, and we can actually quantify that with the PacBio.
Okay,
Yeah.
super cool. Okay, we're going to go back to mic 1, and then we'll go across again. We got lots of time. Thank you, speakers, for staying on time.
Hi, thanks. Charlotte from Netherlands Cancer Institute. I have a question for Dr. Navin and Dr. Curtis, actually, because you guys both talked about early oncogenic mutations in either already normal women or like calculated to be a couple years before the primary breast cancer. One, again, normal women, you don't even know if they'll ever get breast cancer. The other in maybe in metastatic breast cancer patients. So how do you guys foresee predicting which of these early oncogenic mutations will be able to lead to either cancer or stay so dormant that basically the tissue will stay morphologically healthy for an entire life?
Yeah, I mean, I think from our perspective in the normal tissue, of course, what we don't know is, with women that have higher levels of aneuploid cells or higher numbers of CNA events, are they a higher risk for developing breast cancer or not? We weren't able to really investigate that in our study. You need normal breast tissue samples, and then women that develop breast cancer 20 to 30 years later. So those are kind of hard studies to do. But I also didn't talk about a lot of the events that we see in invasive breast cancers that we don't see in the normal tissues. Things like chromosome 8, MYC amplification, for example. We never saw that in the normal breast tissue. We also don't know about the status of PIK3CA and P53, the two driver mutations. Are those present in these aneuploid cells early on, or do those happen later as well? So I think there's a lot of questions still about how you transition into DCIS or invasive cancer from there.
Yeah, I think it's an interesting question. I mean, the fact that we're all somatic mosaics walking around with these mutations, that there's also competition between those potentially mutant clones, I think is key. So I would maybe separate out the phases. I mean, certainly we do see these complex aberrations in DCIS, but DCIS is a long way from normal, right? And there's a lot of intervening stages. I think personally, I think this is sort of a call to arms to do deeper studies of pre-malignant biology so that we really know what is promoting that risk. And I think, fortunately, there's been some new efforts to understand the molecular basis. We need to do it in the cohorts that have long-term follow-up, and that's intrinsically hard, but I think we can get there. And I think we also have to reconcile the relationship between these alterations and how our immune system surveils them. And we didn't have time to focus on that here today, but I think, this is also a critical piece.
And maybe also, while we've got both of your attention, how about stem cells? So were stem cells included in either of your studies? That was one of the questions here.
So in our study, we did have so the DNA data couldn't really tell us that. The DNA attack coassay data could help us map the lineages a little bit, but really couldn't help us map in very fine detail to what we would consider to be more stem-like populations and things. I think technologies that can do kind of DNA, RNA, coassay sequencing will be able to help address that in the future. And in studies like the Breast Atlas, we did very detailed mapping of kind of the populations within each of those three lineages. And some of those we think are a little bit more stem-like. So I think those are important future studies.
Okay, super, thank you. All right, microphone 2.
Nick Murray from Adelaide, and I've had a win because the first online question was mine, so I get 2. I'm not quite sure if this is for Dr. Navin or Dr. Curtis, but one of you talked about the emergence of monoclonal variants in metastasis after a period of drug selection. So I just was curious, do you see the same variant in more than one metastasis, or is this something that will happen in parallel in different sites with different mutations, or both?
Yeah, I mean, there's good examples of this when we apply endocrine therapy looking at specific hotspot mutations for, let's say, ESR1. Not all of our therapies potently select for that particular variant. So I think we also see examples that might be more general, but we certainly do see, enrichment for specific classes of events. So genes, not necessarily the precise mutational level, and that will depend on, whether it's a driver or tumor suppressor. But, selection is potent, so we can read some of this out. I think, to your point, in the comparison of primers and mets, one thing that would be hugely helpful is larger sample sizes, right? And you could go deeper and say, well, I'd like to compare metastatic sites, but then you need a rapid autopsy program. So I think, our sample size have been inherently limited by the challenge of that question. It's getting better, though.
Super, okay, microphone 3 now, please.
Yeah. The question to Christian regarding structural variants, subtypes in breast cancer, and potential mechanism that drives this. So double-strand breaks actually present in a normal even cells and carry on through the cell cycle, right? So it's accumulation of those double-strand breaks stochastically in different parts, right? So would present the challenge for the tumor cells and NG2 before onset of phase, right? So, and this is probably the driving mechanism. So that's inability to deal with this double-strand break that would result in, for example, fusions and different other mechanisms of chromosomal instability. So could you comment on this? And if this, you can detect, for example, extra pieces of DNA could be found in the cytoplasm, and STING can interact with that. So this could be merged both of your talks. Okay.
Yeah, I had a little bit of a hard time hearing you from here. I don't know, Reuben, if you're able to repeat, sorry.
Well, I was going to ask you to paraphrase it, but I think it boils down to the source of the break, and is the break then fueling the instability, which is also fueling
It says,
potentially
yeah.
sting activation, right?
It's a sporadic presence of double-strand breaks accumulated in a tumor cells. That tumor cells cannot deal with that due to the different constraints. So, for example, amplification cycling in E1 driving through this, so there's no time, and so on and so on. So
Yeah, but I
accumulation,
would-
sporadic, double-strand breaks.
Yeah, but I would argue they can tolerate it quite well, and maybe you could comment too. But it's a chicken and egg question, right? Where is the initial stress coming from that leads to that trigger? So,
Yeah, I mean, I think this is part of what we're trying to understand. Obviously, we're looking at tumors that already have undergone some of these mutational processes. We know, for example, that ApoBec mutagenesis is episodic, right? Thanks to work from Reuben. And so how early is this? Is it seen in these normal cells as well? Is that the initiating point? We're just exposed to it. But what we see evidence for, I mean, what I was really trying to point to is that there's a tight interplay between how the cell deals with elevated levels of transcription at, for example, estrogen receptor binding sites, recruiting, and we have these kind of duality of mechanisms, right, where we have our loop that are localizing and you have single-stranded breaks. Of course, these things can be repaired. So I think it's a chicken and egg problem. We can't solve it with patient data, right? We need the right models. The question is, if this is ER-driven, which are those models? And there's many in the room that have debated this, right? Of course, we can do some of these experiments in vitro, and I think, but the interplay with estrogen signaling is quite important, and I think we struggle as a field with some of those models that really potently recapitulate that. So there's experiments to be done.
yeah, I would reiterate that it has to come back to model systems to answer some of those specific questions, because you've got overlapping processes that can contribute to breaks and instability and to signaling. So it gets, and that's why a multiplex, a multidimensional analysis is actually really cool to approach it ideally in a clonal system that you can control. Mm-hmm. So but thank you. And don't, come back and feel free to harass the speakers after. It's good. I mean, ask gentle questions. Microphone 2. Wait, she was here first, then we'll go to 1, then we'll go that way. I'm keeping track.
Okay, I have 2 questions. The first one is for Dr. Curtis. Just to clarify, because I think I missed it, did you say that polyclonal seeding was uncommon in untreated metastases?
Yeah, so I was saying that polyclonality is more common in the untreated ones, that we're missing some of it because so many of the Mets that we look at have seen prior therapy, certainly when we go from the sort of adjuvant setting of breast cancer. And of course, there are de novo metastases. We had very limited numbers there, but we see a reduction once they've seen treatment. They look more clonal.
Okay, thank you. My second question is for Dr. Navin. So you mentioned earlier that when you conducted univariate analyses, you found that only age was significantly associated with the frequency of aneuploid cells and CNA events. I was wondering if it would be meaningful or it makes sense to conduct a joint analysis and see like variable interaction, and if so, if you've already done that or if you have ideas to do so in the future.
Yeah, I will say what I try to say is that in the univariate analysis, age was the only thing that was correlated. Actually, in our multivariate analysis, we also had ethnicity and some pathological features as well. We really need larger cohorts of patients, I think, to have good statistical associations with all the metadata variables. So.
Great. Thank you.
Thank you. Now microphone 1.
Thank you. So speaking of the model systems about the breast cancer, I think I have a question for the Christina. Do you know, did you see any like SBS signatures that you see in the ER positive or HER2 positive breast cancer that are enriched with the extra chromosome like DNA signature, right? Because you said enriched with ER signature as well. Should we work on the ER fusion mutation model or BRCA mutation model or p53 mutation model? Like, can you comment on that?
Yeah, I think you're asking if we see specific mutational processes or signatures linked to these focal copy number amplifications that sometimes occur on ecDNA?
Yeah.
Did I get it right? Okay. Yeah, absolutely. So there's a lot, and couldn't get into all the nuances of it, but it does appear to be stereotype, and they track together. But as Reuben pointed out, there's multiple processes that are operative simultaneously, right? So it is hard to parse which came first, right? I mean, you could say a subset of these have chromothripsis as well. We see APOBEC, right? There's, I didn't talk about HRD. You can come to a talk tomorrow if you want to hear about HRD, but to this point, the ones that are mutually exclusive are, for example, HRD and the extra chromosomal DNAs. We don't really see those co-occurring. They represent different axes. And, you asked a little bit about BRCA. Of course, we know that BRCA2 carriers tend to enrich for ER-positive, HER2-negative subgroups. Some of those patterns are seen there. And when we see HRD-like signatures in that subgroup, they look like the BRCA2 ones, not BRCA1. So there's absolutely signatures. And I think, we can go back and try to also relate this to what we see in specific experimental models to give us a clue, but we have to kind of go back to the beginning. And you might have to actually, if we're asking about the question that was just posed, engineer it from the start, right? So I think it's a question, is how well do they relate? We now have a lot of patient data.
So there's a couple of questions related to what we're just discussing here. One is, how do ecDNAs form in the first place? And I find it very interesting that they form in non-triple negative, so non-HRD. And that's actually counterintuitive because one would actually expect them to be higher where there is genome instability and opportunities for looping out by non-homologous end joining. So what do you think the mechanism is?
Yeah, well, I mean, I think some of this comes back to these focal, I think, we see very different patterns. There's a lot of genome instability, but it's not specific. It's genome-wide. You have massive loss of heterozygosity accompanying, right? Like swaths of the genome that are gone. But you don't have these really focal events. And so coming back to that, I mean, I actually think key transcription factors play a role in this elevated transcription. There may well be, and we didn't get into this, so to me, it's sort of intuitive that we see it there. I'm curious. I understand your take on that, but I think it's interesting when you think about the role that transcription plays and the evolutionary mechanisms that may be at play to buffer this by excising a segment of your linear chromosome onto a circle. Yeah. And what we didn't really speak about is that, of course, they can also reintegrate into the genome, sort of at will. And so many of these then appear as HSRs, and it's very hard to deconvolve that from sequence data, right? So absolutely important, and we could come back to the role that other processes, micronuclei, et cetera, play.
Yeah, no, absolutely. And so building on it a little more and maybe to egg the other speakers on as well. So what proportion of ecDNA, so in the HER2 amplification, for instance, it seemed like some of them were chromosomal, some of them ecDNA. It looked focal. Did you get a feel for the sense there, or whether that's dynamic as well?
Yeah, I mean, we can estimate the proportions, and HER2 has the highest. There was other recent studies looking at this, so it's about, it's like excess of 70% of the HER2s show evidence for a circular amplicon. And it's a little bit lower, 60, in excess of 60 for the ER-positive high risk. So a little bit of a difference, not huge. But I think what's interesting is that the complexity of those amplicons also differs. So HER2, we think of HER2 alone, it might, but it travels with other oncogenes, both on chromosome 17 and others. So then there's a question of how are these cooperating, and are they forming like transcriptional hubs? We also didn't talk about
common ecDNAs.
Yeah.
Okay, that's really cool. So this gentleman has been very patient. Mm-hmm. Thank you.
2 quick questions. Christina, you were talking about ecDNAs, and over time you see it connecting to other parts of the genome. Do you think this is evidence of integration and re-excision, or do you think it's pre-existing clones, or is there a way of sorting that out?
Yeah, it's a brilliant question. We do see some new additional events that seem to crop up later. I think the challenge is having really the number of longitudinal samples to parse this. It's a big problem. So we're sort of looking at either unpaired samples and what the proportions are that these co-occur, and sort of a game of frequency. I think the data from the neoadjuvant study, and that's why I included it, is kind of provocative because you can see after single cycle this massive drop. And yet there's other cooperating events. And HER2 seems to like to crosstalk, but it is the highest amplified, is much higher than its friends. So I do think it could be evidence for reintegration. These are complex. I mean, mapping these is hard. Of course, long read data may help us a little bit. But we've been able to go into experimental models and capture those out and show that they really are circles. And I think I just want to stress this point because doing these predictions from sequence data is hard, right? And ground truth is hard. So yeah, there's a lot going on there. The only other point that I'd make is that I think what we see in a cell line model in terms of reintegration under these very different selective pressures may not mimic what we see in vivo. So we can do those studies, but I'm not sure. To Ashley's point, context is important.
Quick follow-up to Ashley. Amazing work with the SAT-seq. Have you gone back to look at whether localization is different in some of the peaks that like highly disruptive, mutations?
Intracellular localization? Yeah, so we see a lot of our constitutively activating mutations enriched in METs, and they're trafficking and degraded, right? So they transit from the ER to the Golgi and then degrade via autophagy-like degradation in lysosomes. So we definitely think that's a key role. And I think our most pro-metastatic variant alters degradation, which is coupled to its trafficking, right?
Excellent, really neat. Coming back to microphone 1. Don't worry, we didn't forget. And I'm going to overrule that thing down there. We actually, because these guys roll on time, we have about 6 more minutes for questions.
A follow-up question for Christina about the polyclonal metastases. It's very interesting because it also supports the potential biological importance of circulating tumor cell cluster-mediated metastases. I just wonder from your analysis, do you see like potential timeline of those polyclonal? Are they both coming or multiple clones come to metastatic sites and like similar timing, or they might be consecutive, like clones, like maybe one arrives first, another arrives later?
So we cannot infer that really from these data because we're doing we're analyzing retrospective sequence data and we're asking about clonal composition. So it's inherently limited. We really can't resolve the timing. We can just say they're distinct. But, and Ashley might be able to comment on this, I think this is the perfect setting where murine models can actually help us understand that metastatic cascade in far greater resolution. And of course, it's also exciting to think about some new tools to sequence CTCs to better study that, because I think, there are ways to go between these systems. I don't know if you want to.
Yeah, I mean, definitely in vivo with barcoding strategies. What we've seen, though, that complicates that is like with STING remodels the microenvironment, right? And so there can be kind of a field effect, too. Like, so a particular variant that creates an immune suppressive microenvironment can promote growth of other variants, too. So that's, I think, a factor to consider. If it's a extrinsic effect, you might not want to do that in a pooled setting.
Super. All right, back to mic 2.
Yeah, thank you. So first of all, I want to congratulate you guys. The whole room was full when I walked in, like 5 minutes, your session was closed. It's very hard to see after post-COVID, like everybody wants to be on the, virtual. So congrats. So my question is for Christina. Christina, the question is just like now I'm setting the stage to talk to you later. We have in our cancer center a large number of longitudinal samples from liquid biopsy, if you will. Now, we want to set up a biomarker, for either diagnosis, treatment response, so on and so forth. And we are like inundated with number of things we can do, exosomes, CT, DNA, now the EC DNA. So what do you think is the best way to approach so that we don't miss out on something rare, but also capture, you're getting where my question is at, like how will you set up and study which, if you have access to a longitudinal array, like people who never cancer, no cancer, in the beginning, got cancer, got treatment in the survivorship program, what do you think is the best way to do, thinking about it? Sorry, it's $1 million question, I guess.
No, it's a good question. It's a big question. I think we have to also separate what we can use right now to guide clinical care as to determine treatment, to monitor, for example, minimal residual disease and change course. And so I think, there are tools out there to do that. They're constantly improving sensitivity-wise and also the kinds of readouts. I think there was a session earlier today, or maybe it's tandem with this one. So there's certainly, new kids on the block for measuring what we can from the periphery, in a non-invasive manner. And I think that the dynamics is very powerful. Obviously, being able to do it non-invasively affords a greater opportunity. I think we also need, though, to relate what we see in the periphery back to what we see in the tumor, and we do need some more studies on that to make sure we're not missing things. So I think if it's a research question, of course, there's many things you can do. If it's a clinical question, I think we need to, be very clear on where we're using this information. Happy to chat offline. It's probably a longer answer.
Okay, excellent discussion, and I want to thank everybody here for hanging on. Last question.
So, Christina, somewhat provocative question. As you have shown in colon cancer, there's a prominent clone and there may be smaller clones associated with it. So the question comes is, if we are using Oncotype and all of those assays on primary tumor, and if they are polyclonal metastasis, are the de-escalation therapies that we are talking about, based on Oncotype, MammaPrint, and all the other assays, are they going to be dangerous in the long term? Or what are your thoughts on it?
Yeah, that's also a big question. I would say, I think we need to see how well we can predict recurrence beyond 5 years. And of course, there may be additional ways to do this beyond 21 genes. But I think this is about getting risk stratification right to guide how we monitor and treat patients. And, we've come a really long way, so that's great. I think there's more that can be done. If our risk prediction models are accurate, then, I think your question gets mitigated. This is less of a concern. But if we're talking about a risk in 5 years, but that's persistent beyond that, we may face other challenges. So, we need better tools. We need to better stratify. And we probably need to better monitor and not just sit and wait to see what happens.
Okay, I think, would our speakers like to have any final comments? Other than please come up and visit and ask more questions. Let's thank our amazing speakers one more time. Enjoy the meeting.